Hello and welcome to our webinar today, Catching Up with CRISPR, hosted by Wiley and the American Society for Microbiology. My name is Megan Angelini. I'm the Managing Developmental Editor for ASM Books, and we're so pleased that you can join us today. This webinar is going to highlight the current opportunities and challenges for this disruptive technology and the related societal and industrial implications in medicine, biotechnology, and agriculture. So please allow me to introduce today's speakers, CRISPR pioneers and editors of the book, CRISPR, Biology and Applications, Rodolphe Barrangou, Erik Sontheimer, and Luciano Marraffini. Dr. Barrangou is the Todd R. Klinhammer Distinguished Professor in the Department of Food Bioprocessing and Nutrition Sciences at North Carolina State University, co-founder of Intellia Therapeutics, Locus Biosciences, and Cilia Biosciences and Trico, and editor-in-chief of the CRISPR Journal. Dr. Sontheimer is the Pillar Chair in Biomedical Research and professor in the RNA Therapeutics Institute at the University of Massachusetts Chan Medical School, an associate editor of RNA, co-founder of Intellia Therapeutics, and a scientific advisory board member at Tessera Therapeutics. And Dr. Marraffini is the Caden Family Professor and head of the Laboratory of Bacteriology at the Rockefeller University, a Howard Hughes Medical Institute investigator, and co-founder of Intellia Therapeutics and Eligo Biosciences. We're so happy to have them here for this webinar. So without further ado, let me hand it over to our speakers.
Maybe I'll go first. So good morning, good afternoon, maybe good evening to some of you. Again, my name is Rodolphe Barrangou. I'm a professor at NC State, and I've had the great pleasure and privilege, along with my fellow editors here and CRISPR pioneers, Luciano and Erik, to work on CRISPR for almost 20 years. As a matter of fact, my first CRISPR patent will expire next year. I'm not sure whether it makes me a CRISPR dinosaur or a CRISPR pioneer or both. Time will tell. But it's been a remarkable, enjoyable journey. And to contextualize to some extent, why I'm here, why we're here, I started working on CRISPR from a non-canonical angle. I was working at a company called Danisco at the time, now DuPont, that worked on selling dairy starter cultures that ferment milk into yogurt and cheese. And my task was to understand how and why some of those bacteria survive that process and are great, and why occasionally some of those bacteria are susceptible to bacteriophages and die. And that started my journey with CRISPR. And as a matter of fact, as outlining the book in one of the chapters that I co-wrote with one of my colleagues and students, we showed that the biological function of CRISPR-Cas in bacteria is actually as the adaptive immune system. That's where it started. That's how it was first applied, first commercialized, first deployed, first used industrially. As a matter of fact, for over 10 years now, people throughout planet Earth have been consuming fermented dairy products that were cultured using CRISPR-enhanced starter cultures that are resistant to phage. So if you had one bite of yogurt, one bite of cheese, if you had pizza last night or cheeseburger last weekend or nachos or anywhere in the world, you've consumed a product that was fermented using CRISPR-enhanced starter culture. And of course, as we all know, and as we cover in the book, ever since the world has changed, and a very inclusive collective of CRISPR scientists, including many pioneers who have co-authored chapters in the book, have investigated and determined the mechanisms of actions of CRISPR-Cas immune systems, and we cover their diversity, and we'll discuss that to a high extent here. But outside of their biology, as we all know, CRISPR-Cas immune systems have been technologized. And I and my colleagues have been part of a very colorful, very momentous, very interesting, very exciting journey to use those different CRISPR-Cas technologies for applications and diverse applications, almost as diverse a set of applications as we have a set of CRISPR-Cas immune systems, to deploy them in the context of medicine, in the context of food, in the context of ag, in the context of biotechnology and beyond. So if you're interested in all those topics, or if you're interested or familiar with one of those topics where you're curious about more, there's a lot that we're going to cover. There's a lot that we discuss in the book, and there's a lot that we're going to present today to contextualize that. But there's some irony in CRISPR starting in bacteria, making its way to organisms across the tree of life, and now having an increased appreciation for actually the diversity and the role of CRISPR in bacteria across microbiology for ASM, as a matter of fact. So I'll stop here and give a chance to my fellow editors to introduce themselves and highlight how they came into CRISPR originally and how it's changed in the past few years.
Yeah, I think maybe I should continue, because I think it's a little bit of a continuation of Rudolph's story. Way back in the day when I needed to, if I was finishing my thesis and needed to find something interesting to do, I decided to take a look at CRISPR and decided to start working a little bit on that. And then, of course, I needed to find a lab to do my postdoc, to work on CRISPR, which is what I decided. And that's when I joined Erik Sontheimer's lab. And then together we demonstrated that kind of one of the first molecular studies of the molecular mechanism of CRISPR, showing that it actually targets DNA and not RNA. To put it in context, in the mid-2000s, it was very in fashion to the RNA interference was a mechanism that was in everybody's mind, and actually the Nobel Prize for was given to Mello and Fire for in 2000s for the discovery of RNAi, and the first bioinformatic predictions of CRISPR was the missing RNAi in bacteria, because the machinery for RNAi is in archaea and in eukaryotes, but it's absent in bacteria. So there was some sort of idea that CRISPR will work similarly to RNAi and cleave RNA molecules, which with time, then some of the diversity that Rudolph indicated of CRISPR systems turned out to be that some of them actually do target RNA molecules, but the majority, and also the ones that are important for biotechnological applications, actually they cleave DNA, and that's what we demonstrated with Erik, and I'll let him tell more about that. My second kind of important contribution or key moment in the development of the CRISPR field was through a collaboration with Fang Zhang, where we were one of the first groups to transport the CRISPR-Cas system from Streptococcus pyogenes and put it into human cells, and show that using Cas9, you can introduce both indels, deletions or insertions, as well as site-specific mutagenesis in human genes, and which a paper that, along with another from the Church Lab that was published back-to-back with us, kind of started the gene editing of human cells and then gene editing of organisms. So I have the privilege and pleasure to work again with Erik and also with Rudolph. We know each other for a long time, and edit this book that will, in a minute, I'll cover a little bit of what the book covers, but it's the biology and the technology. And yeah, it's been great to have all the contributors, very important people in the field that we have to acknowledge them, that although they're not here, but it's very important to say that the book will not be possible without them. And I'll leave now Erik to introduce himself.
Thank you, Luciano, and it's a pleasure to be here and to join my fellow panelists. I came into the CRISPR field in a different direction. My background was more focused on RNA science, and mostly on pathways that regulate and execute gene expression in eukaryotic organisms. And so we had been working on messenger RNA splicing for a while. And then we were very active in the field of RNA interference, which you just heard a mention of from Luciano. And so my entry into the field was really very much arose from my two co-panelists here. The CRISPR pathway first hit my radar screen in March of 2007 from Rodolphe's work, where they effectively showed that it was a genetic interference pathway that confers adaptive immunity in bacteria. And because we were just about the entire lab was focused on RNA interference, that caught my attention, but we hadn't been working on bacteria in a long, long time. And so it was when Luciano contacted me about joining the lab as a postdoc that I really started to think seriously about jumping into this field. And as you heard from Luciano, he came to my lab, which at the time was at Northwestern University in Evanston, Illinois. And we started out with the work that he already described on the fact that CRISPR has a fundamental distinction from RNAi pathways in that it can operate by DNA destruction. And this is because we had known what was happening in terms of technology and therapeutics development with RNAi. This is when we first alerted the field to the possibility that the CRISPR system, if we could understand better how it works and if it can be ported into eukaryotic cells, would confer an RNA-guided DNA targeting system that could potentially be extremely useful. And as we all know, that has in fact come to pass, and it has really been an amazing thing to watch develop. But I think it's a beautiful illustration about how different scientific directions, whether it be from biotechnology, food science, basic microbiology, and then gene regulation and RNA, all converged very beautifully in the phenomenon known as CRISPR. And at the time that Luciano contacted me, there were roughly 20 articles in PubMed that mentioned CRISPR, and that number now, I believe, is over 20,000. And so it's really been an amazing thing to watch the field expand. So in 2014, my lab moved to the RNA Therapeutics Institute at UMass Chan Medical School in Worcester, and we have continued to work on CRISPR ever since, including Cas9 specific anti-CRISPR molecules, new genome editing platforms, and most recently, the development of split prime editing platforms. And we'll hear more about all of those things as we go through the webinar. Rodolphe?
All right, thank you, gentlemen. So we have introduced ourselves to some extent, provided some contextualization and context for why we're here. And the reason is we got the great opportunity to edit together this book about both CRISPR biology and its many applications, and touching a little bit on the implications to some extent in the context of microbiology and beyond. So what we're going to do right now is over the next 15 to 20 minutes, we're going to walk you through a few slides and excerpts from the book to provide some background so we're all on the same page about CRISPR-Cas systems, biology, diversity, and their applications. And then we'll open it up for Q&A thereafter. And on that note, we invite all the participants to use the Q&A button at the bottom of your Zoom webinar option menu, and we'll field those questions as we go through after we've had a chance to introduce and present some of the content of the book. So without further ado, I will hand the virtual microphone back to Luciano and let him get us started on the outline and the content of CRISPR biology and applications.
Right. So I hope that you all can see the screen, as Rodolfo has a real copy of the book. Here's the cover. So as Rodolphe mentioned, I'm gonna go through kind of what we try to do with the book, and here's a very overview of all the chapters. Basically, we have divided the book into three sections. The first section is about the molecular mechanisms of CRISPR systems. As Rodolphe mentioned, there are many different CRISPR systems. that have different molecular mechanisms of targeting different nucleic acids. And this section covers those at detail. Then the second section is about the biological functions of these different CRISPR-Cas systems. What do they do in bacteria? The different functions. Mainly, they work as an immune system, preventing phage or plasmid infection, but they have also been found to have very other cool functions and abilities for bacteria. And of course, the last section is what CRISPR systems can be used for in biotechnology. And so it covers a wide range of technologies that are based on CRISPR-Cas systems and RNA-guided nucleases that belong to these systems. So why bacteria have CRISPR is because they need to protect from phage infection. And I thought it would be fun for the audience to see here kind of the main reason why bacteria have CRISPR. It's a little movie, and here, here there's the green dots are phages that are labeled with a green dye. So every cell in this, there are 6 cells, and all the cells are being infected. And this is what happens with cells that are infected with phages and don't have CRISPR. They explode. They grow a little bit and they explode. So for that reason, bacteria really need to protect themselves from these predators, these viruses of bacteria, the bacteriophages. And the way CRISPR works is through 2 phases, or mainly first there's an immunization stage, or as Rodolphe mentioned, his discovery that CRISPR constitute an adaptive immune system. And that is because a CRISPR locus is composed of repeats. CRISPR means cluster regularly interspaced short palindromic repeats. In this cartoon, the repeats are the black rhombuses, and these repeats are intercalated by short sequences that we call spacers that come from phages and plasmids. And what we call spacer acquisition, the ability of CRISPR systems to collect a little piece of the sequence of a virus, it provides it's collecting information about infection that then is used in a second stage of CRISPR to destroy the incoming virus that is infecting the cell. And so the way that this information in the spacer is used to destroy, to provide immunity, is first through a step of CRISPR RNA biogenesis, which is basically the transcription of the CRISPR array and the processing of that RNA that contains repeats and spacers into the short RNA guides that you all know probably that are used by RNA-guided nucleases present in CRISPR's systems, that then form complex ribonucleoprotein complex with these Cas effectors, and they find complementary nucleic acids, and then in that way they provide immunity. And I think that the most common way, and one that is it's at the basis of technological applications, is what you see here with these scissors. CRISPR have been called also molecular scissors, where they cut the DNA of the incoming phage. But in truth, the recent work on CRISPR have shown that CRISPR can provide actually 2 different types of immunity. And the one that I just mentioned to you, the molecular scissors that cleave the DNA, is what we call cellular immunity. The DNA of the phage enters the cell, is cleaved by CRISPR, and that cell that is infected survives. And that's basically how Cas9 works, which you are probably familiar with. But recent work have shown, especially with the CRISPR systems that target RNA, and they find a complementary RNA sequence to the guide RNA, those provide immunity at the population level. And what I mean by that is that the infected cell actually does not survive. The infected cell is sacrificed within the population in an attempt to prevent the spread of the virus. That the virus enters a cell that enters growth arrest or dies, so that virus cannot propagate and generate more viruses. And that for that reason, the cells that are actually not infected are the ones that come back, and that's how immunity is achieved. And type 6 systems, which is the number indicates, is the last of being found, actually works this way. And actually type 3 systems also work a little bit like that. And it's actually type 3 are a mixture of both of these two types of immunity. And I'll touch upon that later. So as I mentioned, there's a lot of diversity on CRISPR, and I think that Rodolphe is going to touch upon that, right?
Indeed, yeah. Thank you, Luciano. And I think, again, understanding for the audience why those bacteria have immune systems, how they function, but remarkably how diverse those strategic options to defend themselves against invasive nucleic acids like plasmids, of course, viruses, bacteria, bacteriophages, transposons, and the like is very important. And it was mentioned, I think Erik mentioned that one of our goals when we put the book together was to bring to the readership the contributions that a lot of our fellow CRISPR pioneers have made to the field. And as a matter of fact, Kira Makarova, Yuri Wolf, and the unique Eugene Koonin have made tremendous, repeated, several over decades of work, milestone contributions to CRISPR-Cas genetics, CRISPR-Cas biology, CRISPR-Cas nomenclature, which is very important, and was not trivial when we first started the field, as a matter of fact. And they were kind enough to contribute a chapter going through the evolution and classification of CRISPR-Cas immune systems. So as you see here, and as you have heard already, and some of you may know, there is a multi-layered classification system that is based on classes, major types, and subtypes. So we're not going to go to the subtype granularity, given that this is an overview webinar, so to speak. And if you want to understand this, certainly do read that chapter and all the other chapters in the book. But generally speaking, as you can surmise by now, CRISPR-Cas immune systems are comprised of two elements: the CRISPR array, as articulated with the spacer and the repeats, and the Cas, CRISPR-associated sequence machinery, the Cas genes that encode Cas proteins that enable them to have the mechanism of action that Luciano walked us through with adaptive acquisition, cRNA biogenesis, and eventually Cas-dependent interference against DNA and or RNA, as we just heard. So class 1 is defined by Cas machinery that constitutes a complex, right? Such as the CRISPR-associated complex for antiviral defense, the cascade complex. So it's a multi-protein effector complex that carries out the interference, whereas in class 2 systems, we have the convenience and the portability of a single signature effector interference protein. So class 1 system encompasses type 1. We just heard about type 3. There's still a little bit less characterized type 4. And then the class 2 systems are arguably the most famous and infamous and used and widespread and popular because of that portability of that single effector Cas signature protein. For type 2, this is the infamous Cas9. For type 5, it is Cas12. And for type 6, it is Cas13. And if you go back to the original literature, you may realize that until 2011, those genes had previous names. If you've heard of CSN1 or others, or CSC and cascade complexes and CMRs and others, that nomenclature has changed over time. And as a matter of fact, every few years, Kira, Eugene, and a number of our colleagues get together to update, upgrade, and contextualize from an evolutionary standpoint, genetic standpoint, and biological standpoint, the different classes, types, and subtypes. And it's very important for CRISPR microbiologists, obviously, to understand what CRISPR-Cas system they have and how it works and what it does, how it functions with the biology aspect. But of course, it's very important for the toolbox, because that CRISPR-Cas system genetic and mechanistic diversity has been the very basis for the discovery of new effectors and for the technologization of new effectors that we'll go through application in a few minutes here. The other important aspect here that is well covered in this chapter is the amazing and remarkable occurrence and widespread diversity of those different systems across many diverse phylogenetic units of bacteria and archaea. As a matter of fact, in bacteria, those CRISPR-Cas systems occur somewhere between 42 and 46% of the phylogenetic units and the genomes that are available out there. And in archaea, it's somewhere between 86 and 90%. So those CRISPR-Cas systems are widespread. They're a dominant immune system that bacteria use across the tree of life that we see here. That being said, though, type 1s and type 3 are the most dominant. They are more complex. They are genetically, mechanistically, maybe a little bit more sophisticated and interesting. Of course, type 2 systems are very popular, but they're not as widespread. They are the third most widespread CRISPR-Cas system nonetheless. And this is why types 4 and 5 and 6 were characterized later on and subsequently, because those are not as dominant and they're more rare relatively. So there's a broad diversity of CRISPR-Cas immune system that occur in a heteroscedastic manner across phylogenetic units of bacteria, but they are widespread nonetheless, and they collectively provide DNA-encoded, RNA-mediated nucleic acid interference, whether it's DNA and or RNA. So read Kira and Eugene's chapter, and they articulate that in a very compelling and interesting way. Next slide, please, Luciano. So as some of you know, there's a lot of excitement around type 2 systems and the CRISPR-Cas9 systems because of their technological use. As a matter of fact, the very first work that Luciano and Erik alluded to that we published in 2007 was based on a type 2 CRISPR-Cas system from the Dairy-Bacterium Streptococcus thermophilus. And we showed at the time that there are 4 genes involved in this locus. Some genes are involved in acquisition, some genes are involved in interference, and some genes are involved in both. And of course, the infamous Cas9 was shown subsequently to contain an HNH nickase domain and a RUFC nickase domain that allows those systems to selectively, specifically, and now we know programmably target and reprogram and redirect CRISPR-Cas systems for sequence-specific targeting. So there are 3 subtypes of CRISPR-Cas type 2 systems, 2A, 2B, and 2C, with 2A and 2C being more widespread and technology used thus far, including some of the pioneering work in Neisseria meningitidis that Erik and his colleagues have done as well. Of course, SPY-Cas9 is infamous, and others like ASAU, STH, CJE, and others are there. So there's diversity within systems, within types, and within subtypes as well. The mechanistic approach is the same: DNA encoded, RNA mediated, in this case, DNA targeting. And there's a remarkable ribonucleoprotein complex comprised of 2 distinct natural RNAs. So Erik talked about the importance of RNA research and RNA scientists early on in the system. There's a tracer RNA, trans-encoded CRISPR RNA, discovered by Emmanuel Charpentier, as a matter of fact, that provides a complex with Cas9 to process the full transcript, the pre-CRISPR RNA coming from the CRISPR array, and then processes those long pre-CRISPR RNAs into single spacer-containing guide RNAs that drive the interference. So you end up having a Cas9 loaded with a dual guide, made half of the tracer RNA and half of a CRISPR guide that is encoding a spacer sequence derived from a single vaccination event to direct DNA, complementary DNA targeting and cleavage by the dual nickase HNH and RUFC endonuclease to provide a blunt cleavage exactly 3 nucleotides away from the 3' edge of the spacer and protospacer. And this technology, as you see on the right, has been repurposed to do all kinds of flavors of genome editing. It's briefly mentioned in section 1, chapter 4, and Erik will discuss that in the application realm in subsequent chapters that are covered after chapter 11. Next slide, please, Luciano. So what's interesting is that mechanistically, early on, there was a lot of work in the early years, right, between 2008 and 2012, to understand how interference worked. And that mechanistic understanding of the genetics, chemistry, and biology of CRISPR-Cas interference and double-strand DNA targeting and cleavage was the basis, as a matter of fact, for the repurposing of those molecular machines for genome editing and more. And the way it works is you have this complex formation, again, as mentioned, two different species of RNA, the cRNA, the CRISPR RNA, and the trans-encoded CRISPR RNA, the tracer RNA, that are loaded into a two-lobed Cas9 effector made of recognition domain and nuclease domain. It forms this Cas9 ribonucleoprotein complex that then scans invasive genetic elements, to look for the immune system target sequence by complementarity. So it's looking for an invader that matches this piece of DNA that is loaded in there, which is derived from a previous exposure and vaccination event. And ironically enough, we discovered in 2008 the PAM, the protospacer adjacent motif, which is a conserved region. Think of a short 2 to 5 nucleotide sequence that is conserved. So there's some homologies to restriction enzymes and NRM systems, ironically enough, for those fans of history of microbiology, molecular biology, biotechnology. And that PAM sequence is first targeted with a PAM targeting domain. And once Cas9 latches on to the PAM, then an R loop is formed to interrogate the sequence from the guide that is loaded and see if there is a DNA complementary match. And if that match is through the seed sequence is a bona fide complementary match, that unwinding of the DNA will extend from 3' to 5' and create an R loop. And upon full formation of the R loop, the HNH domain will nick one strand, the RUFC domain will nick the other strand, and it will generate in the phage and or the plasmid DNA, we showed that in 2010, that it will generate an endonucleolytic cleavage, which essentially prevents viral replication, plasmid replication, and vaccinates and interferes and vaccinates the strain against those invasive genetic elements. And of course, that approach has been repurposed to do genome editing and build a pretty sophisticated toolbox that now a lot of molecular biologists, synthetic biologists, evolutionary biologists, bioinformaticists, and others use to either at the bottom mine and look for novel Cas9 effectors in metagenomes, novel genomes, environmental samples, and the like. And you have to find the effector, and you have to find the PAM, and you have to find the guide RNAs and their structures. Of course, on the top right, we can do directed evolution in the lab setting, sometimes even high throughput, to increase the activity and or the specificity of those effectors. And then thanks to structural insights, as a matter of fact, Cas9 structure is amazing in its own ways, and same for Cas12 and others, of course, Cascade and others, can provide and inform rational design and structure-driven enhancement of portions and or motifs and or residues of specific biochemical interest from Cas9 to make it more specific and manage targeting and or affinity and or activity. So if you're interested in Cas9, type 2 CRISPR-Cas systems, certainly chapter 4 would be of high interest. I think I'm going to hand it back to you, Luciano, to contrast that with type 3s.
Right. So I'm talking about type 3 because this is the what we work in the lab also, and the chapter that I wrote. And I think I'm just going to highlight the other, what Rodolphe showed with Cas9, in essence, that clips the DNA, that allows the cell to survive. And that's the one type of immunity. Type 3 systems can do that as well. In essence, I'm not going to go through the details, but there's also the generation of the guide RNA and the formation of a complex with a guide RNA. But then the guide RNA finds a transcript of the invader in, let's say, the phage. And then things are a little bit different because some transcripts of the phage are produced right away after the onset of infection. And when that happens, the type 3 complex, which is it's called the Cas10 complex, it goes and recognizes that there's a virus, there's an invader inside the cell, and uses the DNA cleavage activity, very much so as Rudolph explained for Cas9, to destroy the genome of the virus, and then the infection is complete, is stopped, and then the cell can continue growing. But then you can imagine also that there are other transcripts in the viral cycle of infection that are produced very late during infection, and when that happens, the cells are already very much compromised by the phage. The cells are there. It's difficult for the cells to come back and regain full viability if they let the phage reproduce and replicate for a long time until actually the transcript of the phage is produced and Cas10 can get to the phage and recognize that there's an invader inside the cell. But when that happens, the Cas10 complex has another activity that we call it the POM domain. But importantly, what it does is synthesizes a second messenger. It uses ATP to produce a molecule that is a ring of 4 AMPs. And that it's a ligand for another Cas protein, another CRISPR-associated protein that we call CSM6, which is an RNase that is a non-specific RNase. It has 2 domains. One is the domain that binds the second messenger, and the second domain is the RNase domain. When the ligand is bound by CSM6, then that triggers the RNase activity. And as I said, it's non-specific, and it degrades all RNase inside the bacterial cell, both the RNA of the phage, and that it's important to stop the phage, but also the RNA of the cell. And that also prevents the cell to continue growing and supporting the phage to grow. Phages, they need a very healthy cell to replicate themselves. When the RNA starts degrading the transcripts of the bacteria, then those cells become sick and they cannot support phage infection. And therefore, in those situations, those cells don't proliferate, the ones that are infected, and the ones that actually survive are the lucky ones that don't get infected. And so I wanted to show you this. We have that in chapter 5, but we also have the next chapter, I think chapter 6 about type 6, not chapter 6, a later chapter about type 6 systems that exclusively do this population level immunity. And to move on, I think that then we need to move on to a very important aspect of CRISPR, which is the applications. And for that, I'll leave Erik to go into that.
Thanks, Luciano. Yes, it's important to note, as we've seen with many other technological revolutions, that it really started with basic curiosity-driven science. And the early practitioners of CRISPR research were not really looking for a genome editing technology, but that's what appeared whenever we understood the biology better. And it's also, I think, very important to note that the CRISPR field has moved amazingly fast. As a parallel that's very much in my mind, RNA interference was first discovered around 1998, and it took 20 years before the potential for therapeutic development became a reality, where the FDA approved the first RNA interference-related drug. And at the time, that was viewed as lightning fast. And what we are likely to see, there are clinical trials ongoing for genome editing therapeutics, but no FDA approvals yet. But it's widely expected that the first approvals by regulatory bodies in Europe and likely the US could come as early as 2023, later on this year, perhaps 2024, but it looks very, very promising. And so that would be within about 15 years of the pathway's discovery and within about 10 or 11 years of its first demonstration of genome editing. And so one reason for this is because the CRISPR technological revolution has coincided with many other technological revolutions, including high throughput DNA sequencing, transcriptome-wide profiling, cryo-electron microscopy, mass spectrometry, many other computational advances in particular. Many other technologies have come around and exploded at roughly the same time, and this has resulted in great synergies that has led to many technologies based on CRISPR. So I don't have to say much about genome editing here because we already heard about type 2 systems, and the crucial piece of it is that it is RNA guided. And basically, you can think of the Cas9 protein or the Cas12 protein as being like a form of hardware, but then the guide RNA is a form of software that allows you to reprogram that hardware so that you can tell it to go here in the genome or go there in the genome. And this has been really one of the keys to its utility and its adoption. And so there are a number of different aspects of CRISPR genome editing. You can use it as a nuclease to introduce double-strand breaks that then get repaired by the cell in ways that can change the underlying sequence. More recently, there have been additional implementations involving the fusion of Cas9 to other proteins like deaminases or reverse transcriptases to make for base editing and prime editing. And all of these have different utilities. There will never be a single CRISPR system that does everything we need it to do. It's the branching out and the building of different tools that allow you to do different things that increases the underlying things that can be addressed by using CRISPR technology. Luciano, next slide, please. So this is further illustrated here on this slide, and at the very bottom, we can see that what we are looking at is eventually going in vivo. Clinical trials are in progress. The first approvals will likely be for ex vivo applications where edits are introduced in cells that are taken out of the body and then reintroduced. This is for various hemoglobinopathies, including sickle cell disease. But delivery still remains the biggest challenge and the biggest hurdle for in vivo applications. And there are many companies and research labs that are working on different solutions for the delivery dilemmas, depending on what the target tissue in the body might be. Next slide. Again, really one of the key defining features of CRISPR is that it is RNA guided. And it is possible with Cas9 as a good example to introduce mutations into the Cas9 protein that still allow it to fold and bind its guide RNA and do just about everything else that we ask it to do, but it will not introduce breaks in the genome. You can inactivate the enzymatic domains that actually cause cuts in the genome. And so this means that it is no longer going to be as useful as the direct genome editor like we heard, but it still retains its RNA-guided DNA binding technology, so you can still make it addressable with different guide RNAs. Furthermore, you can fuse or tether additional domains either onto the protein or onto the RNA in ways that will allow that protein to drive different outcomes, whether it be to induce the expression of a gene, repress the expression of a gene, visualize a locus in a genome. And so by using these so-called dead Cas9s that we can fuse to other things, this has enabled a large number of applications. You can fuse just about anything you want to these proteins and take advantage of the fact that you can address them to a particular site in the genome to drive a particular outcome. Next slide, please. So this only scratches the surface of some of the technologies and applications that CRISPR has been used for. Nearly all of them are covered in the book by distinguished colleagues. We mentioned genome editing in particular with a chapter by Patrick Hsu and Peter Lofty. And then we also mentioned Lei Stanley Chi and his colleagues with their chapter on dead Cas9 systems. But in addition, the book includes chapters on CRISPR screens. This is something that's a research tool, not so much a therapeutic tool, but it allows you to interrogate entire genomes in large scale as a big pooled population for their ability to either gain or lose a particular function. And this has been a tremendous advance in functional genomics to try to identify which genes are important for whatever pathway an investigator happens to be interested in. CRISPR is being developed as an antimicrobial technology because of the fact that it can target and kill specific subpopulations of complex microbial populations by virtue of the fact that you can program it to recognize specific DNA sequences that we've been hearing. CRISPR-Cas systems can be used to provide phage resistance in industrial bacteria, including the food industry, like we heard about from Rodolphe, and his chapter with Avery Roberts goes into this in greater depth. And finally, a really creative application of CRISPR-Cas systems is to use them to record particular physiological events into the genome, so you can go back in later and read out a recording of a particular physiological event that cell underwent. And this is opening up a number of different research paths as well. So these are just a few of the examples of the dizzying range of technologies that have come out of these core features of CRISPR systems, namely that they are RNA guided and that they can use adaptive systems to record exposure to environmental features and others as well, as described in the book. So we'll close out the technology section with that and hand it back over to Rodolphe.
Great. Thank you, gentlemen. I think this gives the audience a much deeper and compelling understanding of what CRISPR-Cas systems are, how they work, how they function, what their diversity is, and how they can be deployed in all kinds of applications and context to do many things, not just change the genome, but the epigenome, the transcriptome, and engineer bacteria and record information. So Erik, you mentioned the speed at which the field has changed. But in the context of biology, there's a great question, maybe to both of you, about the timing under which DNA is acquired and the speed, maybe at the population level as a clue, how quick and how efficient or not adaptive immunity is. So can either of you gentlemen tell us a little bit about the time constraints of the immunization process?
Yeah, there's a very good question from the audience about a very, very clever question, very clever observation about whether the spacer that is inserted in the array is used immediately to try to prevent infection. And what we understand so far is no. CRISPR works as the same as a vaccination. When you get vaccinated with dead viruses or dead bacteria or non-infected bacteria. And it's similar in the case of CRISPR. There has to be some other event that stops phage infection for CRISPR to acquire a spacer. Part of the work that we did that maybe is that people will understand because it's familiar with other technologies, restriction enzymes. Restriction enzymes also come from bacteria, also cut DNA. And so we show that the place that restriction enzymes cleave, it's a great place for on the DNA of the phage to acquire spacers from that end. And the act of cleavage of that DNA by restriction enzymes actually kills the phage. And there are other groups that have shown other mechanisms that can stop phage infection before it kills the cell, and that is the way that then CRISPR systems have a chance to acquire a spacer and be immune for the next phage, the next round of infection that comes.
Could you comment a little bit, Luciano, how that happens at the population level, and maybe it's restricted to biofilms or not, and how diverse the types?
Yeah, so I don't think it's biofilms. They have characteristics that may make them more resistant to phage infection in the sense that sometimes the outer cells of a biofilm are the ones that actually get infected, and then they kind of take the burden or are able to take out the phage from the population, and then the cells inside the biofilm, they have, they're protected as a shield, right? that's another way of defense that is at the population level, and it's similar to what I was alluding to, although it doesn't have to do with CRISPR. The way CRISPR does it, it doesn't require a biofilm. Any population in any form can using CRISPR can do this population level immunity.
So we talk about immunity. I know I've highlighted adaptive immunity a number of times. So can we explain to the audience whether it's innate or adaptive from a theoretical standpoint?
I think I also can take a kind of the work basically that demonstrated that with Rodolphe by showing that CRISPR, the cells that become resistant to phage, they actually acquire a spacer. That act itself, it's an acquired type of immunity that allows the organism to adapt to its environment. It's a mechanism of adaptation. For that reason, it's adaptive and not innate. Innate will be something like a restriction enzyme that is not tailored to any particular phage. Any phage that has a sequence that it can be cut by restriction enzymes, then it will be subject to restriction immunity, so to speak. But CRISPR can allow the organism to adapt to new phages, constantly adapt. A new phage comes, then the spacer is acquired, and the CRISPR will have the ability to target it and destroy it.
It's worth noting, I mean, hopefully one thing that has come out from this discussion as well as from the book is just how much the CRISPR field has expanded over time. And very recently, there have been some discoveries of CRISPR systems that somewhat blur the boundary between innate and adaptive immunity. The innate immunity, one of the concepts behind it is that it recognizes pathogen-associated molecular patterns, as the lingo goes, or PAMPs. And that is distinct from the canonical view of CRISPR. But very recently, there have been some examples of things besides DNA sequence recognition that can trigger the activity of new variants and kind of twists on the classical CRISPR mechanism. So just like we've seen with other technologies, the boundaries get blurry as the biological understanding expands.
So there's a question, and maybe Erik, I pointed to you here about concerns that Cas9 may damage DNA, which is kind of how we use it in the first place. So any clarification about how DNA repair pathways come in and why CRISPR is being used towards clinical applications in the context of CRISPR as one part, and maybe DNA repair pathway is another part?
Sure. So the first implementations of CRISPR were to introduce double-strand breaks into the genome. And CRISPR was not the first technology that was formed to do that. Zinc finger nucleases and TALENs came earlier. But again, the fact that it is RNA guided is what made it so valuable. And so it had been known, thanks to the work of Maria Jason and others in the early 1990s, that double-strand breaks can be hijacked, as it were, or co-opted to change the underlying sequence. And that is at the core of the initial wave of genome editing technologies. And so really CRISPR is about introducing breaks, and then the cell takes it from there, and it's the underlying repair pathways that function normally in the cell that will do the editing. And so it's important to note that double-strand breaks happen every day in all of our cells, and that we have evolved mechanisms to deal with them. And so below certain thresholds, they do not appear to be associated with undue damage or collateral effects. But nonetheless, CRISPR's accuracy is not perfect. And so you can end up with unintended sites that are close but not exactly matched to the guide RNA that can still undergo double-strand breaks. And so there are concerns about additional mutations that can happen elsewhere. Furthermore, these breaks can cause translocations where one part of one chromosome joins with another part of another chromosome. This is something that has to be guarded against. And so there's been a great deal of attention paid to on versus off targeting. I think most of us in the field consider that to be less of a problem than one might gather from the popular press, that the accuracy questions are under better control than you sometimes read about. And conversely, perhaps an underappreciated concern happens about unintended outcomes at the on-target site. And so you don't always get just small insertions or deletions. You can get much larger ones that sometimes fly under the radar of the methods that are commonly used to interrogate outcomes. So the accuracy is certainly a concern, and the regulatory agencies care a lot about this. But we understand more and more all the time. The initial indications look very promising in terms of the accuracy of the treatments that are advancing through clinical trials. And so the general sense is that these have been and will continue to be solvable problems.
So one series of questions here about applications in the context of the clinic is also phage therapies. And as a matter of fact, I and Luciano have been involved in companies that are using and exploiting CRISPR-Cas systems to actually weaponize phages and enhance phage killing and have programmability. What we just heard about genome editing, the programmability of the targeting can be encoded in the CRISPR to use CRISPR not as immune systems to defend bacteria against phages, but CRISPR as targeting systems in phages to actually kill bacteria. So given the rise of antimicrobial resistance, given the desirability of narrow spectrum antibiotics that can more specifically, more selectively, more efficiently target bacteria, select bacteria responsible for infectious disease, there's a lot of interest in deploying those CRISPR-Cas systems and delivering those CRISPR-Cas systems as programmable antimicrobials. And some of those pursuits are already in the clinic and will be continuing to get to the clinic shortly. Luciano, you want to expand upon that to some extent?
Yeah, or yes. There's it's an interesting twist. And there are actually phages that do have naturally occurring phages that do have CRISPR. I think that it's an evolutionary race, right? And so. But we, as in the side of applications, it's a very good tool to have. And then speaking of phages, there's also another good question from the audience that says, why diversity of CRISPR exists? And that's a very short answer to that is because phages are very, very diverse. It's the most abundant and diverse biological entity on Earth. So in order to be able to defend against so many different types of phages, bacteria need different types of CRISPRs.
Yeah, and as a matter of fact, one word, evolution. Phages are really good at evolving workarounds, and so the CRISPR system has to adjust. It's a constantly moving target, and that drives ever-expanding diversity in the biology.
Yeah, that arms race, I was going to do that on both sides of viral evolution and host evolution has been mesmerizing and very impactful. And to answer Angela's question, indeed, Eugene and Kira and others have been investigating the evolution of CRISPR-Cas systems, which is also why it's so interesting to see that they've co-opted other effectors, right? Like transposons, like integrases, and things like this that really complexify and sophisticate the ability of the host to circumvent phage escape mechanisms. As a matter of fact, we haven't even talked about anti-CRISPRs, but there's an arms race that includes multiple layers of molecular mechanisms that really push the agenda on both sides of the host and of the virus, and the evolution reflects that. Any last minute question you guys want to address? If not, we'll answer those offline. I see Megan is here. We're at time.
Megan is here, so.
We'll thank the audience for their time. I will thank the editors on behalf of everybody else here and give the mic back to Megan.
Yeah, we're right on time here. So I want to thank Drs. Barrangou, Sontheimer, and Marraffini for the excellent discussion that we've had here today. And we want to thank the audience for all those thoughtful questions and for taking the time to join us today. And with that, we're going to close out our webinar. Thanks so much, everyone.
Thank you.